Okay, good morning, everyone. Sorry for, I'd actually have to do this so that at least I'll save my time. Thinking of traveling, so by the end of this week, we will not be able to finish this, and I've already made my mind to round off this class by the end of this class. So I will give you the assignment I promised to give so that we can, I think I can call the next person to take care of the next class. Okay. So I think we discussed this. I don't need to repeat myself here. We are going to start with activation of amino acids. This is just a two-step reaction that made an amino acid to be ready for addition into a peptide. How does that occur? First of all, the amino acid need to be attached to a transfer RNA, a specific transfer RNA. And I told you the role of DM in identifying the specific transfer RNA for a particular amino acid. Just like we said, we have a specific amino acids, specific transfer RNA for each amino acid, as well as this specific amino acid transfer RNA synthesis. For example, if a methionine is going to be added to transfer RNA, then the enzyme methionine tRNA synthesis will be responsible in addition of methionine to specific transfer RNA that code for the anticodons bearing methionine or anticodon for methionine. Then it will add it to the 3' end. And we told you that the 3' end is made up of cytosine, and adenine. So on the 3' end, the adenine, we have the 3' hydroxyl group of adenine or adenine nucleotide. This is the position where the amino acid will be added. So this reaction need energy, just like we mentioned that see how energy in form of ATP is used to catalyze some of the reactions. So this particular activation reaction is one of them. It's called charging, like charging the transfer RNA with specific amino acids. So this reaction is in two steps. First of all, we have the ATP. We have the ATP converted to AMP by the removal of a pyrophosphate. And the energy release will be used to attach the AMP to amino acid and also to the active site of an enzyme. So we produce an intermediate complex of enzyme, AMP, and amino acid. And this complex will be responsible in identifying the specific transfer RNA by interacting with the D arm of transfer RNA before it catalyzes the addition or attachment of the amino acid to the acceptor. And basically, this reaction involves the reaction between the hydroxyl group, as I mentioned, and the carboxyl group of the amino acid leads to formation of an ester bond. So we have amino acid transfer. I hope that to be clear. Okay. So discussing the stages in protein synthesis, we're going to discuss this process, like I mentioned, in three different stages, starting with initiation, elongation, and termination. And initiation basically is a step where it involves the assembly of the protein synthesizing workbench. And this workbench basically comprises of the messenger RNA, the transfer RNA, and the ribosome. They will come together and package themselves then before the synthesis will start. That is the initiation. While the elongation is the continuous addition of the amino acid to the growing polypeptide. So we have the first amino acid attached to the second amino acid attached to the third amino acid, like this, up to the last amino acid in the protein. This stage involves the formation of the peptide bond, and then we have the termination, which basically involve the identification or recognition of the nonsense codons, the determination codons. Therefore, the synthesis will start. Okay, now discussing the initiation. The initiation, we're going to talk about 4 stages, which involve the ribosomal dissociation, formation of pre-initiation complex, and then formation of initiation complex. So we have 143S pre-initiation complex, then 48S initiation, and then 80S initiation complex. So we'll see how this complex to perform. And under elongation, we also talk about these 3 stages involved. The first of all, binding of the transfer RNA, mean the activated amino acid to the A side of ribosome, and then the formation of the peptide bond, and then lastly translocation, where the next codon on the messenger RNA will be detected for the next transfer RNA to bind. And then we have a termination. Okay, now discussing the first stage, which we said the initiation. Now the initiation, like we classify the initiation, first of all, the ribosomal dissociation. In eukaryote, you can remember that we said the eukaryote have an 80S ribosomes, and this 80S is divided into, it formed 2 subunits. It formed the 40 and 60 subunits. So dissociation of this 80S ribosome will give 60S and 40S. Because these 2, 60 and 40 are not stable when dissociated, because they can easily reassociate and come back to 80S. So the 40S will be attached with the eukaryotic initiation factor 3 and 1A. So eukaryotic initiation factor 3 and eukaryotic initiation factor 1A will bind to 40S, thereby stabilizing or preventing the reassociation with the 60S ribosomal subunit. And this stabilized 40S will now combine with a ternary complex. And this ternary complex is nothing but eukaryotic initiation factor 2 bound to GTP. And in addition, it bound to a ribosome transfer RNA that was already attached to methionine. And you remember methionine is the first amino acid that will be synthesized, because the start codon is AUG, and AUG code for methionine. So the signaling is the identification or the starting of the protein synthesis start with the AUG, which is the start codon and code for methionine. So first of all, we have the messenger RNA transfer RNA that is being charged with methionine already. So this eukaryotic initiation factor 2 bound to GTP become active, and then it bind to the transfer RNA bound to methionine to form a ternary complex. And the ternary complex were now attached to the 40S subunit to form 43S pre-initiation complex. So this binding of initiation factor 2 to GTP is a kind of, binding of the initiation factor 2 bound to GTP to the methionine bonded transfer RNA is a kind of regulatory step so that this protein itself, the eukaryotic initiation factor 2, can undergo phosphorylation, dephosphorylation reaction, of which its phosphorylation can actually prevent the binding of the GTP, which leads to inhibition of this process. So it's a kind of regulatory point. We'll talk about another regulatory point as well. So we have 43S pre-initiation complex. The next thing is this 43S pre-initiation complex will now bind to the messenger RNA containing cap-binding protein, or stabilized cap-binding protein. How this stabilized cap-binding protein is produced is here. First of all, we have the messenger RNA that comes from the transcription. And after all the modification splicing and the rest, we have the messenger RNA with 5' 7-methyl guanosine and 3' polyadenine polyatal. Okay, so we have this initiation factor, eukaryotic initiation factor 4G, 4A, and 4E that comes together to give eukaryotic initiation factor 4F complex. The 4F complex is what is referred to cap binding protein. So the cap binding protein will now attach itself to the messenger RNA to the 7-methyl-guanosine cap, and this complex is unstable until when the initiation factor, eukaryotic initiation factor 4A and 4B comes and bind to the messenger RNA, thereby stabilizing. And binding of 4A and 4B required energy in form of ATP. So we have this messenger RNA complex now binding to 43S3 initiation complex to form 48S pre-initiation complex. Remember, we said formation of 48 initiation complex. But what we have here is 48 pre-initiation complex, because first of all, there is a need for the arrangement of this transfer RNA on the messenger RNA. Remember, the transfer RNA coding for methionine or bonded already to methionine, like we just mentioned. And we have the codon for methionine on messenger RNA being AUG. So we have an anticodon for AUG. So first of all, an energy is needed for the transfer RNA to scan over the messenger RNA and relocate this position where the AUG is located on messenger RNA so that it can hybridize. So what exactly happened and energy is needed for this reaction where ATP is now being utilized for that, number one. Number two, also, how does the messenger RNA transfer RNA actually identify this AUG? Because remember, there are quite a number of AUG. AUG can be found, so methionine can be found at the second, third, fifth, etc., because it code for methionine, and methionine can easily found in the protein sequence. So there is a sequence called Kozak consensus sequence. That particular sequence on the messenger RNA determine the position where the AUG is usually found around that consensus sequence. So even if you get AUG along the messenger RNA and it's not around it, there is no Kodak consensus sequence, then that AUG might not be the actual start coder for the messenger RNA synthesis. So usually that sequence Kodak codon is actually identified, and then the AUG will be found somewhere around like 3 to even 4 sequence upstream and downstream can be found somewhere around. So once the transfer RNA bind to it, then that is the start codon. In the case of prokaryote, the Shine-Delgado sequence is actually the sequence instead of Kozak consensus sequence. So that leads to formation of 48S initiation complex. And the next thing now is binding of this 48S initiation complex to 60S ribosomal subunit. And that leads to the release of this eukaryotic initiation factor 3, 1A, etc., including eukaryotic initiation 1, 2, which is bonded to GTP. But before the release, there is hydrolysis of the GTP leading to GTP, and then the eukaryotic initiation factor 2 will have to be recycled by interacting with another eukaryotic initiation factor 2B, and that leads to the action of a kinase where the GDP will be phosphorylated to form GTP, and then you have eukaryotic initiation factor bind to GTP, and then also another eukaryotic initiation factor 2C will now help in interaction between the methionine transfer RNA to eukaryotic initiation factor 2 bound to GTP. So the process will continue. Of course, like quite a number of these ribosomes are needed for protein synthesis. So also we have this ribosome with the help of eukaryotic initiation factor 5. We have the 48S initiation complex will now bind to the 60S ribosomal subunit to form a complete ribosome, which is 80S initiation complex, ready for the synthesis of protein. And we can see we have the ribosome, we have the messenger RNA, we have the transfer RNA coding or carrying the first amino acid, and we already identify the first start codon using the Cossack consensus sequence. Now the next thing is, remember this transfer RNA carrying methionine is placed in P site. So it occupies P site where the transfer RNA bonded to peptide will be found. So we have a site being free still. So the subsequent transfer RNA bringing activated amino acid will now bind to this A site. We'll see how it's happening. Okay. Now the next we have elongation stage, and in the elongation stage we have binding of amino acid transfer RNA, and then formation of peptide bond. Just like I started explaining, like the first transfer RNA bound to the P side. Now we have another RNA, tRNA, that will bind to the A side. Specifically now, the messenger RNA, the next codon will be read, and that will identify which amino acid need to be added. So remember, we already have this activated amino transfer RNA, activated amino acid bonded to a different amino acids ready. So once it is detected by the ribosome, the next codon will be detected. Now the next codon will come and complete and bind to the ribosomal RNA with the help of TUC arm, where it binds, and then the anticodon will complement. By complementing, the next thing now is the formation of. So this binding is energy requiring process. So it uses energy in form of GTP, and then eukaryotic initiation. Elongation factor 1 alpha will facilitate the interaction between the amino acid transfer RNA, the new coming amino acid transfer RNA with the ribosome, and then the binding to the A site. Binding to the A site will now bring the 2 amino acids. The first one, which is the methionine, and then the next codon amino acid. Any amino acid could be valine, could be threonine, could be serine, phenylalanine, etc. So the next thing now, we have seen the proximity between the 2 amino acid. Now the next is formation of peptide bond, and we already know how peptide bond is formed between the carboxyl and the amino group. So what simply happens is that the carboxyl group that form an ester bond with methionine in the peptide side or P side will now cleave with the help of the 60S ribosomal subunit. And the 60S ribosomal subunit has this enzymatic activity of peptidyl transferase. So this is a ribozyme, an enzyme that is RNA enzyme. It is RNA, it performs a function of an enzyme. So it cleaves this ester bond and then facilitates the formation of peptide bond between the amino group of the next amino acid, the new amino acid, and the methionine to form a peptide bond. And once the peptide bond is formed, you can see that the first transfer RNA that was involved in assembling the ATS pre-initiation complex will now be empty. So the next thing is we have translocation. And in translocation simply involves the movement of this ribosomal subunit, the initiation complex, ATX initiation complex. So it will move. When it moves, it will go away from the first codon, the AUG start codon, because the amino acid has already been transferred to the next transfer already. So once it moves, then the hybridization will be uplifted, and uplifting that will move the transfer RNA specifically for methionine to the E side, the exit side, and that will lead to its own release. And then the movement of this second amino acid to the P side of the ribosome. So we have the A side to become vacant. So the A side will also be occupied with a new codon, which code for another next amino acid that need to be added. Then the amino acid, the transfer RNA coding for that amino acid will come and bind, and then the process continue of bond formation, peptide bond formation like this, and then the ribosome also continue to move it. That is why we have the structure of polyribosome, where you have more than one ribosome being attached to a particular messenger RNA. One particular messenger RNA having more than one ribosomes, and each of the ribosome is carrying out the protein synthesis simultaneously. So we have the termination. So this continue until when the stop codon is reached, the nonsense codon. So once the stop codon is reached, and there is no any specific transfer RNA for that, and then we have the release factor will come and bind. The binding of release factor will facilitate the binding of GTV, and this help in facilitating the breakage of the release of the newly synthesized protein with the help of peptide transferase. So that once it is released, the protein newly synthesized protein will be released. If the ribosome will continue for the synthesis, so that's the end of the synthesis, and that will also trigger the release of the subunits. Will have the 40S and 60S will release, so that the protein synthesis has been concluded. And the next thing, we already have the peptide. We have the nucleotide link, amino acid link to another amino acid peptide, a long chain that need to be assume a secondary structure before its on activity. So we have quite a number of post-translational modifications, starting with folding. First of all, the protein need to be folded to assume the secondary conformation, and that secondary conformation makes the protein to even be active. And we know that folding is basically by the role of chaperones, and these chaperones, they are of different classes. We know of because chaperones are heat shock protein, and this heat shock protein, we have heat shock protein 70, we have heat shock protein 60, we have heat shock protein 10, all different classes. So heat shock protein 70 basically involved in the folding of protein, and also the chaperoning system. With the chaperoning system, which is made up of the heat shock protein 60 and 10, they come together and they involve in folding of the protein. So please, you can read more on the protein folding also. And then we talk about proteolytic cleavage. So some enzymes and even proteins that are produced in form of inactive form, like zymogen, and which need to be activated. The inactive protein, I think we can remember trypsin and trypsinogen, which they trypsinogen. So they are proteins, they be released in form of inactive. A particular condition that leads to cleavage of a particular segment will now make the inactive protein to be active. Therefore, once it is active, the activity of the enzyme can actually start. So the next one, we have intent splicing. We have intent and extent, just like in exons and introns, where some intervening sequence of amino acids are actually not necessarily for a given peptide, therefore need to be removed. So once they are removed, so the fragment can, the extent will now come and join together to give a functional protein. So lastly, there are covalent modification, which involve phosphorylation, glycosylation, methylation, etc. And at least we talk about methylation and phosphorylation. Glycosylation even also more especially when we're talking about glycoproteins. So some glycoproteins will added to, some proteins will have a conjugation or will conjugate with a sugar or a carbohydrate to form a glycoprotein, mostly found on the surface of the cells. So it's all the things. So part of the basic post-translational modification before the final activity of the protein. So you can read more on this covalent modification as well. So I have these 3 to 5 textbook you can consult. You can read more on basically on what I discussed in the class, right from the subcellular component to this stage. And I'm sorry, I think I have to add another one here. I have an assignment. So you divide the class into 5-6 groups then. One will take gene expression in prokaryote, one will gene expression in eukaryote, one will take gene mapping, the application of gene mapping, and another one will take functional genomics, and another one will take the application of functional genomics. So please, this is the 4, let me put it now. Let me see if I can put it now. 5 functional genomics. 6 and genomics. So what am I expecting? I'm expecting you guys to make a notes on this. Remember, you see, most of my lecture notes contains a lot of, in fact, basically contains the structures, illustration, etc. So this is basically more important, although you will add some note in it, but that note should be back up with all this illustration. And, each group should have a note that is not more than, let's say, 45 pages, not more than 45 pages for this, but make sure you've done justice to each of the topics. So for per group, I should have like 4 to 5 students, so that at least 6 group can cover the whole class. Yeah, even if it means 3 students, so we can have this. Okay, so please, I don't know, I would have done that if I am with you. So please try and do that so that each will carry a particular group. I will create, what do you call this? A Google form that you fill this thing per group. You fill this, but you still have to create a binded copy of this. So each group should have a binded copy of the assignment, not more than 5 pages. Submit to me. So I think with this, then we'll see by the next class if there is anything I need to come to see you. If not, then I will let the next person come and continue with the class. And if there is any question, I know you can reach out to me so that we can discuss.